So in this video, we're going to continue our discussion of DNA replication in eukaryotes, but we're going to look specifically at how replication proceeds in the telomere region. So of course, as we remember, eukaryotic chromosomes are linear. And so at a point, you're going to reach the ends of these linear chromosomes, which are these telomere regions. And we need to see how replication goes here. So what's important to remember is, of course, DNA replication via polymerase proceeds from the 5' to the 3' direction on the daughter strand. That's how it's synthesized. And when we look, though, when we get to this template strand, so this top strand here that I've drawn is our template strand. This 3' region over here is called the 3' overhang. That is our telomere region. It's the end of this chromosome. So we're slowly adding on DNA in the direction of the arrow. We're building this way, like that. Well, once we get to the end of the chromosome, we come across two problems. One is that, of course, our DNA polymerases cannot place a nucleotide freely. We cannot just take a DNA nucleotide and add it here. There must be a pre-existing strand there for a DNA polymerase to act on. And then secondly, there is not enough room to add an RNA primer to then synthesize DNA off of. So we need to figure out some way to replicate this 3' overhang region so that our chromosome isn't shortened on each subsequent round of replication. And that's where this enzyme telomerase comes in, which is shown here, this red circle blob thing. Now, telomerase has two important components. It has what's called a TERT RNA sequence, and it has two TERT subunits. Now, what these refer to, or the TERT subunit, is referring to telomerase reverse transcriptase. So if you understand what reverse transcriptase means, that gives you a hint as to how this process is going to go down. So what's important is that the telomeres are made up of these repeated sequences shown here on this top template strand of this repeated TTA-GGG sequence kind of over and over again. So what we notice in this telomere sequence, or in this telomerase enzyme, is that you have an RNA sequence that's going to be complementary to this repetitive sequence of the telomeres. So it contains this RNA sequence here, which is shown. And as you see, this AAU-CCC region is complementary to the telomere repetitive sequence. So that's going to allow this telomerase complex to bind complementary to so you're going to bind in that CCC is going to bind to that GGG region, which then allows you to, let me erase this 3'. We can then use the RNA subunit here, or this RNA sequence, as a template to then lengthen this 3' overhang. So we can then add a TTA GGG here, which is exactly what we would want, because we want to continue lengthening this. Once that happens, then we will translocate this telomerase enzyme over here, and we can continue building. We can add another TTA GGG. And we can continue doing that until our telomere is long enough. So let's say that is long enough now, but you would continue doing this until you've lengthened it a significant amount. And what that allows us now is you now have enough region or enough space to add an RNA primer to this extra region. You can now build an RNA primer there that will then allow DNA polymerases to come in here and fill in this gap with our normal primase and then DNA polymerase, ligase activity, things like that, endonucleases. All of that normal process will then occur once we have this process happening. But that's how we resolve this issue. And then, of course, you would digest this RNA primer after, and you're left with another 3' overhang, which you would then use in the next round of replication. So, of course, what happens is, eventually over subsequent generations, our telomeres will shorten. And what that can lead to is something called senescence, or lead to cells being senescent, something like this. And what that is just essentially after our telomeres become short after subsequent replications, they act as kind of a buffer to protect our DNA. But of course, as they become too short, cells are going to lose their ability to divide. They're going to die off a little bit. This is kind of what happens as we age. This is one of the reasons. And it could be potentially that our telomerases lose their activity, or maybe that there becomes a lower concentration of telomerases in the cells. But that's kind of the mechanism of how telomeres are replicated in eukaryotes. I hope this video was helpful, and we will see you in the next one.